Good morning. Greetings from Cambridge, Massachusetts. My name is Laurie Boyer, and I'm a professor in the biology and biological engineering departments at MIT. I'm happy to have the opportunity to talk with you today about some of our work as a recent awardee of the FlexSite 96 research grant. So my lab is very broadly interested in understanding the mechanisms that drive cell fate decisions during mammalian heart development and how faulty regulation can lead to disease. Today, I will focus on two very short vignettes. One is a story where we identify functionally how a transcription factor called TEAD1 controls cardiac conduction. And at the end, I'll just spend a few minutes talking about how we've developed new protocols to now use patient-specific iPS cells to also investigate the pathophysiology and function of cardiomyocytes in congenital heart defects. So I'll focus today's talk on a couple of projects, as I mentioned, but I would be remiss if I didn't say none of this would be possible without the work from Vera Koldova and Kirsten Schneider, who really, even despite a pandemic, were able to do some fantastic work and make progress using the instrument for addressing key questions relative to interest of our lab. So in order to understand the factors that drive cardiac cell fate, our lab has focused a lot of attention in the past on transcription factors and how they bind to transcriptional enhancers, as shown as an example here on the left, to control tissue-specific gene expression patterns. So for all of you who don't think about enhancers every day, essentially they're non-coding DNA regulatory elements that comprise a number of motifs that are bound by transcription factors at specific times and places during development. And it's this constellation of transcription factors that communicate with a neighboring promoter to drive gene expression. And so to this end, we and others have identified, using approaches that are often called genomic approaches, we've identified a number of transcription factors that can bind to active cardiac enhancers. And these include the who's who of transcription factors, NKX 2.5, GATA4, MEF2A, and so on. But with in addition to these known and well-studied transcription factors, we've also identified other transcription factors that co-occupy active cardiac enhancers, but their roles are much less understood. One example is TEAD1, which is shown here, which is a T family of transcription factors, and it often co-binds active cardiac enhancers. And it was shown 25 years ago to have essential roles in heart development. Using a gene trap model in mouse, investigators showed that TEAD1 caused embryonic lethality around day 11. And although the hearts appeared to form, they noticed that these animals had thin ventricle walls and that there was a reduced number of trabeculae within the heart as well. Strangely, not much has been done with TEAD1 in terms of really understanding its function in heart development since that time. So we thought for us it's a good place to start in trying to uncover novel mechanisms that drive heart development. So we needed a system. Because TEAD1 levels increase in cardiomyocytes themselves during heart development, we designed a method that would allow us to conditionally deplete this transcription factor during a dynamic stages of differentiation from embryonic stem cells to cardiomyocytes. So shown here is just a schematic of an assay that we typically use in the lab, where we can coax embryonic stem cells from the mouse into mesodermal, cardiac progenitor, and finally a cardiomyocyte fate. In order to be able to deplete TEAD1 in a conditional manner in this dynamic model, we turn to some modern tools where we use CRISPR-Cas9 to insert a degron tag at the 3' region of the endogenous locus of TEAD1. So we were able to use these homozygous ES cells, and by the addition of a small molecule compound called DTAG13, which targets this tag for proteasomal degradation, we could show that we could conditionally ablate nearly all of TEAD1 in a matter of hours. So with these tools in hand, we then asked, what was the role of TEAD1 during cardiomyocyte differentiation? For our purposes and what I'll be discussing today, we added the compound prior to the cardiac progenitor stage at a time where TEAD1 levels start to increase. Surprisingly for us, we did not observe differences in the proportion of cells that represent not only progenitors, but also cardiomyocytes. So FACS analysis showed similar proportions of both of these cell types in the TEAD1 depleted cells. However, we noticed a very unique phenotype where we could tell that these cells at the cardiomyocyte stage at day 10 beat quite erratically. And of course, maybe that's not surprising because that the knockout animals still form their hearts. They just showed some slight changes in the overall ultrastructure of the ventricles. We did note, however, that this abnormal beating looked more like an arrhythmic phenotype, and so we were curious to understand what else that might mean. But it's rather unsatisfying, as you can imagine. It's subjective to describe any kind of phenotype just simply as these cells beat irregularly. And so it was, luck would have it, it was around this time when the lab received the FlexSight CardioExcite instrument. Because it's important for us to quantify the measures or the consequence of T loss during our dynamic differentiation protocol, Vera and Kirsten would really need to optimize the conditions, including finding the right cell number, appropriate media, type of scaffold used, et cetera, in order to really come up with a protocol for robust on-plate differentiation to these 96-well CardioExcite and FlexSight plates. It wasn't as simple of just putting cells on these plates and seeing what happened. We had to allow these cells to undergo the process of differentiation at the same rate that we normally do in a different culture system. So after a lot of trial and error, they were successful in doing so. And I'm just going to show you a key piece of data here that was very exciting to us. Around day 8 of the protocol using the CardioExcite plates, you can see here that we observe an increase in the amplitude of the contractile amplitude. Sorry, these are on FlexSight plates, and that this amplitude increases until the end of the protocol at day 10. And these are, I'm sorry, this was also mislabeled. These are actually the control cells here, where the DTAG cells, which are in blue, showed no similar increase. So with that in hand, we felt we are now in a good position to understand how these cells were affected by the loss of TEAD1. And I want to say also that there's one important advantage of this protocol. It allows label-free and non-invasive measurements of cardiac function, which was really great for us because we could still use the cells at the end of the experiment for downstream assays such as metabolic measurements and immunofluorescence. So we have this assay in hand, and then we wanted to ask whether or not we could quantify this irregular beating phenotype. So what you're seeing here on the left is our work on plate differentiation using the FlexSight 96. And I'm showing you here data from day 10, because I just showed you in the previous slide that we see this maximal difference in contractility at day 10 between the wild type and the mutant. So we first differentiated our DTAG cell lines on these plates. Measured contractile amplitude at day 10, shown on the left here, is the mean beat amplitude display of control, represented by the black line, followed by the T1 depleted cells, represented by the red line. The dotted lines represent standard deviations from the mean beat amplitude. So just for clarity, the mean beat is the average response of all the individual cellular contractions during a 10-second sweep. The inset shows a representation of the individual beats during this sweep. So control cardiomyocytes exhibited a regular beating frequency of about 0.5 hertz, which is shown here. And unlike that, and high amplitude, we observed a dramatic decrease in the D-tag treated cells, which is shown again in red. These data are also summarized in this figure, where you can clearly see a significant decrease at day 10 in the amplitude. So we were also able to calculate beat rate regularity covariance as shown here in this diagram. So this beat rate regularity is a measure of the distribution of interbeat intervals. And so a value near 0 represents regular rhythmic beating, or obviously the higher value that we see for the T1 depleted cells means that these cells beated rather irregularly, which is exactly what I showed you in the video. So again, strikingly, we observed this deviation, and these data suggest that T1 leads to a highly variable beating pattern. But seeing is believing. So I just wanted to show you some videos of what we took on the actual flex site plates. Here are the controls on the left. Oops, apologize, the movies. And then the D-tag depleted cells on the right. And on the left, you can clearly see that these cells beat quite well, which is reflected in their ability to deflect off the membrane and give us the measurements we just saw. However, the D-tag depleted cells really showed this strange pattern that Vera likes to call the disco ball effect. And as you can see, it seems that the rhythmic beating has become uncoupled between cells. And so we're pretty excited to see these data. And we wanted to then ask a little bit more information using the Excite 96, which involved taking the extracellular field potential. So essentially, we performed the same on-plate differentiation using the cardio Excite and using EFP, which is a proxy for action potential in cardiac electrophysiology. We show here in the left panel that EFP amplitude for the control and D-tag treated cells in black and red respectively were quite different. Notably, the early depolarization phase of the EFP is vastly attenuated upon T1 loss. And the repolarization phase is also significantly smaller in amplitude, as you can see here in this inset, which represents the EFP traces during spontaneous beating. So the pattern is also quantified here, and you can see that the median of the absolute values of EFP amplitude are also strikingly different, where we see a greater amplitude in the control compared to the D-tag treated cells. So we also analyzed EFP regularity, and note here that the control cells show high EFP regularity, where again, the D-tag treated cells show much greater variance. So to us, these data suggested that T1, not only did it affect the contractility of the cardiomyocytes, but it also affected cardiomyocyte electrophysiology. So in a lot of data that I'm not going to have time to tell you about, we basically came to the conclusion from quantification of these very important functional characteristics that loss of TEAD1 led to dysfunctional cardiomyocyte automaticity and contractility. And this was actually a huge, breakthrough for us, because prior to using the Nanion technology, we're very unclear the true nature of the defects that we were observing just with this irregular beating. So given this dramatic shift toward the binding of TEAD1 at enhancers, the fact that we observed this change in function, we next wanted to begin to understand what the downstream consequences on a genetic level could be from TEAD1 loss. So we began to take a closer look at the possible pathways during cardiomyocyte differentiation. So as I mentioned at the beginning, TEAD1 has been shown to bind to enhancer sequences that can activate key cardiac genes. This is a simple heat map that represents ChIP-seq data for the binding of TEAD1 at specific sites at the genome. The dark color means that a region is bound. And here I'm two modifications for histones that also mark active enhancers. And the long of the short of it is we can identify binding sites for TEAD1 in both cardiac progenitors as well as cardiomyocytes. But interestingly enough, it appears that when the binding shifts towards cardiomyocytes, that TEAD1 definitely binds more to active enhancer states. So the next thing we did is we wanted to determine what happens to the actual genes. We can use enhancer predictions to predict downstream genes, and it appeared that the neighboring genes to these enhancers seem to play a role in a myriad of cardiac functions, including contractility. But which genes were deregulated by loss of TEAD1? I'm showing you here by RNA-seq. The genes in red are significantly deregulated, either upregulated or downregulated. And when we examine the roles of these genes on a broad level using gene ontology, again, we see that loss of TEAD1 leads to changes in gene expression for genes that function in pathways include cell adhesion, contraction, sarcomere organization, as well as morphogenesis. So this was very exciting results because it suggested that our data at the genomics level were in really high agreement with the functional studies that I just showed you. So based on also the pathways that seem to be disrupted in these cells, predicts that there could be problems with sarcomere organization or structure. So we next wanted to examine the sarcomeres in these cardiomyocytes. And here's just a cartoon of the contractile unit of the cardiac muscle, where here these Z discs represent a single sarcomere unit. And there are a number of proteins that you can stain for or perform immunofluorescence that can give you sort of this striated pattern, and it can give you a sense of how well the sarcomeres form. We performed this assay, and as you can see here on the top, in control cells, Hooks stains the DNA, actinin, which is one of these proteins that are found within the sarcomere. You can observe these nice kind of stripe patterns that we saw in the cartoon. This told us that in the control cells, at least, that good sarcomeres were formed, which is not surprising given the robustness of the contractility and electrophysiology measurements in these cells. However, when we depleted TEAD1, we saw something quite different. So the actinin patterns were much more disorganized, as you can see here, suggesting that the cardiomyocytes didn't form quite properly. We also took a measurement, which we call the Z-line interdistance, and plotting here on the right, the median continuous Z-line distance, we can also show that loss of TEAD1 led to shorter sarcomeres or shorter sarcomeric units, which is in line with this fact that we were seeing them be more disorganized. And notably, shorter sarcomeres typically would be expected to lead to a reduced force of contraction, again, consistent with our flex site and cardiac site measurements. So finally, what I haven't showed you yet or told you yet is that we noticed that there were a couple of genes, and one gene in particular, that were strongly downregulated upon loss of TEAD1, that this gene became deregulated at the cardiac progenitor stage, but was completely lost in cardiomyocytes. And this gene is called VKM1, sometimes known as CD106. And it was really interesting to us for several reasons. So VKM1 has recently been shown to be used as a reliable marker along with cardiac troponin T to enrich for cardiomyocyte populations. And also it plays a central role in a number of cell types, including cardiomyocytes, in cell adhesion, and its disruption can lead to loss of cell-to-cell contacts as well as signaling. So we wanted to then ask whether VKM1 was localized to these sarcomeres. And as I'm showing you here in the top panel, in control cells, we see nice overlap between VKM and actinin. Actinin, again, marks the cardiac sarcomeres in the cardiomyocytes. In contrast, upon depletion of TEAD1 using the DTAG small molecule, again, we see a loss of VKM1 here that appears to, again, be a problem for organization of these sarcomeres. So again, this was really exciting to us because it suggested that perhaps VKM1 might be a direct target of TEAD1 and that we could have identified a new axis in regulating cardiomyocyte function. So in order to test this specifically, we then wanted to block VKM1 in otherwise normal cells, and we asked whether or not we observed the same phenotype as loss as TEAD1. So in control cells here, again, staining of actinin shows these nice sarcomeres. However, blocking VKM1, which is typically can be on the surface of these cells using an antibody, we could see some disorganization of the sarcomeres. And as shown here on the right, similar to loss of TEAD1, again, we saw shorter sarcomeres. So, again, this was really exciting data for us because it suggested that we had identified at least one gene that was a direct downstream effector of TEAD1 in our cells. But then given that, we also expected that loss of VKM1 should show the same type of functional defects as I showed you a little bit earlier. So I'm just going to show you one piece of data here for the interest of time. And so in these cells, we measured impedance using the cardio-excite. We again performed the on-plate differentiation. I'm showing you data at day 10 of differentiation. The blue traces are from control cells, where the dotted lines, again, are standard deviations from the mean. And as you can see here, control cells show really nice impedance, whereas these measurements are severely reduced in the DTAG lines, which are shown in gray, as well as inhibition of VKM. So these data suggested to us that TEAD1 acts upstream in part to VKM1, and that VKM1 is critical for cardiomyocyte cell-to-cell coupling. And we already know that this electrical signaling and coupling between cells is critical for sarcomere maturation, which is in line with our phenotype of seeing more disorganized or less mature sarcomeres. And it's also important, obviously, for propagating electrical signals between cells, which was also read out in our assays. So I hope that you can appreciate that in this particular example, we were able to take a phenotype that we could only just describe as an regular beating phenotype and show that this transcription factor, which really didn't have a very specifically known role, works upstream of a number of pathways and factors that control cardiac function, although didn't per se affect cardiac cell fate. So we are now in a position to publish this work, and it would not have been possible again without the tremendous efforts of Vera and Kirsten, as well as our collaboration with Nanion, including a lot of help from Ron Knox in being able to interpret and analyze these data. So just very briefly, in the interest of time, I also want to give you a progress update on some work that we've done with human iPS cells using the same instrument. So as you can imagine, for anyone who knows about human iPS cells, they're much, much more difficult to culture. They're more finicky. So we again had to work out a number of conditions that allowed for on-plate differentiation. And this work was carried out by Vera and Kirsten, as I showed you. And we're able to plate cells onto the flex site and cardioexcite plates and drive them through multiple stages of cardiomyocyte differentiation, where we ultimately get a nice monolayer of cardiomyocytes. And here I'm showing you that these trisomy 21 cells, which is an iPS line that we focused on, and trisomy 21 is critical for 95% of all Down syndrome cases. The reason why we wanted to study trisomy 21 in heart development, because although it's been studied most commonly for its roles in neurodevelopment, the vast majority of Down syndrome patients also present with congenital heart defects. However, the role of this extra chromosome, or this aneuploidy, in causing these congenital heart defects is much less clear. So we differentiated these cells towards cardiomyocytes, and we have an isogenic control here. And as you can see, I apologize for the noise, but these cells beat quite nicely in the dish at a rate of what we typically see for all normal iPS lines. However, the trisomy 21 cells appear to beat, in this case, a lot faster. So we were a little bit perplexed by this observation at first, but it seemed to agree with some of the measurements we were able to take on, oops, I apologize for that, the flex site plates, where trisomy 21s exhibited a greater mean beat amplitude over time as well. So, and that's consistent with these images. So this is just one piece of data that we have, and we're generating a whole set of measurements to try to better define what's happening in the trisomy 20 lines compared to normal or wild type. But what was really exciting to us is that there's been recent data in the brain that suggests that neural precursors actually appear to mature somewhat faster with the presence of this extra chromosome, which then leads to aberrant maturation and then obviously morphogenesis of tissue structures in the brain. And that may be exactly what's happening in the heart, where we see increased contractility and beating at the same time we see a certain type or slower beating in the euploid cells, and that over time, again, these cells appear to gain the ability to contract, suggesting that they mature a bit faster. So we're continuing this work with Down syndrome, as well as using a number of other patient-specific cell lines. And we're trying to hopefully use not only some of the genetically encoded reporters that we've started to generate, but we hope that we have the opportunity to continue to use the Nanion instrument to get down to the nitty-gritty details of functionally how some of these mutations that are known to cause really serious congenital heart defects are able to do so. And combined with our ability to analyze gene regulation at a single cell level, as well as perform epigenetic assays, and our ability to measure metabolic states, this is kind of like the perfect scenario where we should be able to really dig deep into the mechanism of how some of these congenital heart defects are manifested. And the hope is really to be able to identify some key vulnerabilities that might be used for treatment in the future. So with that, I want to thank lab members that were responsible for this work. I mentioned Vera and Kirsten are largely responsible for all of the work on protocol development, as well as the measurements that were shown throughout this talk. Kunle Demiran, a former graduate student, produced the genomics data. And again, none of this would have been possible without the really generous help from Ron and from our many discussions with Elena. So unfortunately, because I'm giving this talk remotely, I won't be able to answer live questions, but I'd be happy to discuss protocol development, interpretation of any of our results, as well as future directions if you have any questions. And I imagine the organizers will forward these questions to me. So thank you for your attention and for patiently working through this remote presentation with me. So thank you.